Hi all and welcome back to the video module on biological nitrogen fixation. In the previous video lectures, we have studied in detail about non-symbiotic nitrogen fixation. Under this, we studied two organisms, mainly Azotobacter and Azospirillum. Azospirillum, though, is not all normal or conventional non-symbiotic nitrogen fixing organism. It comes under a group of organisms that show associative symbiosis. Under symbiotic nitrogen fixation, we have done two organisms, that is Frankia and Rhizobium. Frankia is an actinomycete that is found in association with the actinorhizal plants, and it forms nodules on the roots of the actinorhizal plants as is shown in the figure here. Rhizobium too is a symbiotic nitrogen fixing organism that shows root nodule formation in the roots of leguminous plants. So in today's class, let us study about the mechanism of symbiosis, taking an example of Rhizobium. Now when we talk about the mechanism or establishment of symbiosis by any organism, here particularly we are talking about the establishment of symbiosis or mechanism of symbiosis by Rhizobium in legume plants. Now when we talk about this mechanism, broadly it can be classified or studied under four steps. Initially, the organism, that is Rhizobium, has to recognize a suitable host. Not all species of Rhizobium can attack or can form nodules with every species of plant. They are extremely species specific, and that is comprising the first step of establishment or mechanism, that is recognition of a host. Once the host has been identified or recognized, the organism or Rhizobium will bind to the root hair of that particular plant, or it will attach to the root hair of that particular plant with the help of several chemical signals. Once it has attached to the root hair, the organism will now start invading, or it will start entering the plant through the root hair and spreading further or colonizing further. It keeps invading, it keeps moving further into the tissues of that particular root and forming a colony over there. Once it has invaded inside, then comes the last step, that is nodulation or formation of root nodules. These are the four steps that are seen in case of Rhizobium and in case of Frankia. The only difference is in Frankia, the chemical signals that are released in these two steps are different. That is the only difference, but the mechanism is similar. First, the organism should recognize the host, it should bind to the host, start invading the host or going further and colonizing further, and finally form root nodules. So let us study each one of these steps in detail. The first step is recognition of the host. So Rhizobium will normally live freely in the rhizosphere of a soil. They do not fix nitrogen, but they are free living organisms. They can survive in the soil freely without being in association with any plant. When there are root exudates that are released from a particular plant, or when certain chemicals are released from a particular root of a plant or a specific host, those root exudates will attract the Rhizobium towards the root. Now, what are these exudates comprising of, or what do these exudates contain? They contain amino acids, biotin, flavonoids. They can contain certain other vitamins like thiamine. So there are several components, one of the major components being flavonoids, which attract the Rhizobium towards the root. Hence, these are acting as chemoattractants. So we have several components, several chemicals that are present in the exudates of the root, and when these are released from a specific host, they attract Rhizobia towards the root. The flavonoids also have one more purpose. The flavonoids that are released by the roots have another purpose. They also activate the Nod genes in the Rhizobium species. Now, if you can recall from the previous classes, when Nod genes are activated, that leads to the production of Nod factors. And if Nod factors are being produced, that leads to the formation of nodule, or it initiates the nodule formation. So, these flavonoids that are released by the root or that are released as chemoattractants not only make the rhizobia come near the root or move towards the root, but also they activate the nod genes of the particular rhizobium species. So once the root exudates have been released and once the organism is now moving closer to the root hair, now comes the next step, that is attachment. Now attachment of bacteria to the root hair is with the help of certain chemicals or certain compounds. So the host plants along with flavonoids, they also secrete lectins. Lectins are secreted by all the species of legumes. Some species of legumes also secrete trifolins. So the plants secrete lectins and trifolins. These are the chemicals. Lectin is a protein and trifolin is a polysaccharide. So these lectins and trifolins are the chemicals which mediate the binding with rhizobium or which help the rhizobium to bind to the particular plant root. So in the second step, that is attachment of rhizobium or bacterium to the root hair, the host plant secretes along with the chemoattractants. It also secretes certain compounds like lectins and trifolins which help the rhizobium to bind to the surface of the root, which means these compounds, lectins and trifolins, act as surface receptor on the plant cell. So to these receptors, the rhizobium will come and bind. Now these molecules, whether it is lectin or trifolin, has common site for both host plant and for the capsular polysaccharide which is found on the surface of the bacterium. So they act as a bridge, or they act, you can see over here, they act as a common factor for binding of the bacteria. So the bacteria which are present will come, the surface polysaccharides of the bacteria will interact with the lectin that is released by the plant root, and now this lectin has a common surface receptor, or it is having common site for attachment of both host, that is plant, as well as the rhizobium or bacterium. There is also another compound which is seen only in rhizobium and bradyrhizobium, that is called as ricadicin. Now ricadicin, you can see the spelling over here. Ricadicin is also a very important compound. It is a calcium binding protein which binds to the calcium complexes on the root hair surface. So ricadicin is also helping in combining or binding the root hair to the rhizobium. So what all is helping in binding? We have lectins, there are trifolins, and we have ricadicin which help in binding the surface or capsular polysaccharide of rhizobium to the surface of the host. So these are acting as receptors which are helping in attachment. Now once the attachment or binding is done, the root hair starts curling. So initially the root hair is linear, slowly it starts curling, as you can see in the diagram over here. So the root hair curls in response to the nod factors that are released by the rhizobium. So let us review this process again. First, there are some root exudates that are released by the plant. As soon as the root exudates are released, rhizobium which is present in the rhizosphere identifies these root exudates and starts moving closer to the plant. Once it moves closer to the plant, the flavonoids that are present in the root exudate will trigger the nod genes of rhizobium. Now when these nod genes are triggered, they produce nod factors which help in nodule formation. Not only nodule, when we say that it is helping in nodule formation, it means it is also helping in curling of the root hair. So we have nod factors which are initiating the nodule formation. Along with that, or at the same time, we have lectins, trifolins and ricadicin which help in binding of this bacterium to the root hair. Now once the binding is done, due to the nod factors that were released, there is curling of root hair in response to the factors which are being released. This curling leads to formation of this kind of a structure where only the tip is curled and the rest of root hair is remaining linear. This type of a root structure or this type of a structure is called as shepherd's crook. So the root hair curling leads to formation of a shepherd's crook structure. And why does this curling happen? It is because when the nod factors are being released, they stimulate, the nod factors released by the bacterium stimulate the plant to produce auxins and cytokinins in the host. So the plant is now going to produce more of auxins or more of cytokinins, which means the levels of auxin and cytokinin will peak in the host, which causes elongation of the root hair and curling of the root hair. So all of these processes are interconnected. First the bacterium is attracted. When the bacterium is attracted, the Nod factors are being released. When the Nod factors are released, they help in again making the plant produce more of auxins, more of cytokinins, which leads to formation of a curled structure that is called as Shepherd's crook. Now once the Shepherd's crook has been formed, once the Nod factors have caused the root hair curling, we come to the next step, that is invasion and nodulation. You can see over here the organism has bound to the rhizobia, has attached in response to the root exudates, it has come nearby and it has attached in response to the chemical signals that have been released. These chemical signals or chemoattractants include flavonoids, but the binding takes place with the help of lectins, trifolins and ricadicin. Once it has attached itself to the root hair, due to the Nod factors getting stimulated, Nod factors then in turn stimulate the plant to release more of auxins and cytokinins, which causes the root hair to curl. This is what we have studied till now. Now coming to the next step, that is invasion. Once the root hair curls and we have formation of this Shepherd's crook structure, now the organism starts invading or the organism starts entering the root hair. Now when it starts entering the root hair, there is a hypha-like structure or a filament-like structure that is formed, which is called as the infection thread. So the rhizobium now forms this infection thread and it enters the root hair through this infection thread only. If you can see over here, the infection thread, the organism has deformed the root cells or it has depolymerized the pectins which are present in the cell wall of the root hair, and it has now entered the root hair. This root hair is like a long channel or like a long tunnel, and this is called as the infection thread. The long tunnel or channel is called as the infection thread. The infection thread is lined by cellulosic or cellulosic substances of the plant, and within it you can see are the bacteria. So the bacteria here are represented by black dots, and you can see over here as the infection thread keeps moving interior to the plant, as the infection thread keeps growing, the bacteria are transported through this tunnel or through this channel or through this infection thread towards the root cortex. So they reach the root cortex only through this channel that is called as the infection thread. So first, recognition of the host followed by attachment of the bacterium to the root hair. Once it attaches to the root hair, we have formation of this kind of a structure which is called as Shepherd's crook. Once the Shepherd's crook structure has been formed, we have formation of infection thread. Infection thread is basically when the cell wall has been depolymerized and now there is a hypha-like structure which is being formed in the deformed root cells. Though the structure is still lined by cellulose or hemicellulosic contents, inside it, it is hollow where the organism can move around. So the bacteria are embedded inside the infection thread, and the infection thread keeps growing till it reaches the root cortex. Now once it reaches the root cortex comes the next step, that is nodulation. So nodulation is seen only in the roots of these plants. We are talking about rhizobium here. So it is seen in the rhizobium plant and it is seen in the root hair or in the roots. There are some organisms like azorhizobium, which is a different species, different genera. In azorhizobium, there is also formation of stem nodules, but generally most of the time in rhizobium or bradyrhizobium or cenorhizobium, in all the different species, we see nodules only in the roots. So these root nodules are now going to be formed. Now how does that happen? So once the organism reaches the cortex, the root cortex, or once it has reached the interior part of the root hair, that triggers hyperplasia. Hyperplasia is rapid cell division. So this hyperplasia is triggered when the bacteria reach the root cortex, because cortical cells or the cells present over there are nearer to the vascular bundle, and this provides bacteria with more or better conditions or optimum conditions for growth and proliferation. That is why the hyperplasia takes place or rapid cell division takes place. Not only that, the organism starts growing. You can see over here, the organism will start growing, and as the organism grows or as the organism proliferates, the cortical cells are also stimulated to divide and form nodules. So the cortical cells will now divide rapidly, or that is what is called as hyperplasia. They divide rapidly and they form nodules. Now, the rhizobium will multiply rapidly and transform into swollen, misshaped or differently shaped or pleomorphic branched forms. These pleomorphic or misshaped branched forms are called as bacteroids. So bacteroids, you can see the terminology over here. Bacteroids are rhizobium itself, but we call them as bacteroids because unlike bacteria, they are now extremely pleomorphic, they are misshapen, they are swollen or enlarged, and this is seen only inside the root nodule, or this is seen only within the root nodule. So inside the root nodule, the rhizobium will multiply rapidly and transform itself into the bacteroid structure. Now what happens to the plant cell? So within the nodule, the rhizobium is enclosed within a membrane. Every cell that is dividing, you can see over here the cells are dividing and they multiply so much that the entire structure gets enlarged to appear like a nodule. This is how the nodule will appear. A swollen, bulbous, enlarged structure which is projecting outside the root. This is happening because of the excessive multiplication of the root cells. But within the root cell or within the nodule that has been formed, the rhizobia are enclosed either singly or in groups. They are enclosed within a further membrane, and that membrane is derived from the hypertrophied tissue of the host. So whatever tissue has been broken down, from that itself a membrane is derived, and inside this membrane we have either one rhizobium or a group of rhizobium that are present. Inside this membrane is what I told you about formation of pleomorphic bacteroids. So inside every cell of this nodule there is further membrane. Inside the membrane there are either one or more rhizobia which are pleomorphic in nature and now which are being called as bacteroids. This membrane or this structure where we have several membrane bound rhizobia inside a cell, that is called as symbiosome. So symbiosome is the structure which is present inside the root nodule wherein rhizobia are enclosed in a membrane that has been derived from the host itself. So the bacteroids within the membrane inside the cortical cells is what we call as a symbiosome. In contrast to the free living rhizobia, these bacteroids are unable to utilize the sugars, and after the formation of symbiosome only the nitrogen fixation will start. So till as long as it is in the form of rhizobia, nitrogen fixation is not happening. Once it enters inside and once it forms a nodule wherein there is a separate membrane that is being formed. You can see over here, this is an infected cell. This is the membrane and you can see the bacteroids inside that particular membrane. So there are membranes inside which bacteroids are present. So these bacteroids inside this membrane is when we call it as a symbiosome. And this is the step or this is the stage where nitrogen fixation can occur or nitrogen fixation starts. And when the plant dies or the nodule deteriorates, bacteria are released back into the soil. And bacteroid, the structure bacteroid which is present inside the symbiosome itself is incapable of division. But there will always be a few dormant rod-shaped rhizobial cells which are present in the nodule. So when a nodule or when a plant dies and the nodule starts deteriorating, these dormant rod-shaped rhizobia will proliferate. They will be released into the soil. They will proliferate and initiate infection in the next plant, or they will maintain a free living existence. See, like I told you at the beginning, rhizobia can survive in a free living manner in the soil. It is just that when they are found in the free living manner, they cannot fix nitrogen. So once the plant dies and the nodule is deteriorated or has broken down, the rhizobia are released and they either continue to live freely or they attack the next plant. They go and infect the next plant. Now it has been seen that in rhizobia, the host specificity is very, very important, like I have told you earlier. So it is extremely specific to the type of host that it is going to attack. This was the mechanism of nitrogen fixation in rhizobium and legume plants. So first is recognition of the host, which is with the help of components like or chemoattractants like flavonoids, biotin, thymine, amino acids, etc. These flavonoids will not only attract the rhizobium, but they also trigger the nod genes in rhizobium, which are now going to help in nodule formation. The second step is attachment. In attachment, we have the help, or the rhizobium takes the help of ricardicin, lectins and trifolins, which help the rhizobium to bind to the root hair. Once it is bound to the root hair starts curling to form a structure called as the shepherd's crook. Once the shepherd's crook has been formed, we come to the third step, that is called as invasion, wherein the hypha is, now there is a hypha like structure called as infection thread that is being formed in the shepherd's crook, through which the bacteria start entering into the root. Once the shepherd's crook or that infection thread reaches the cortex, then the cortical cells are stimulated to multiply rapidly, and that leads to the formation of an enlarged structure which is called as the nodule. Within the nodule, within every cell of the nodule, there will be rhizobia which are bound by a membrane. This is called as a symbiosome. This is where the nitrogen fixation takes place. So this was about the mechanism of nitrogen fixation in rhizobia. These are just to show you the different steps that I have just told you, that is flavonoids attract rhizobium, then we have the help of lectins, trifolins and ricardicin which are helping to bind the rhizobial cell to the plant. It leads to curling of root hair and formation of shepherd's crook. Then there is formation of infection thread. Infection thread, once it reaches the cortex, there is hyperplasia of the cortical cells to form nodules, and inside the nodule we have symbiosome, inside which we have bacteroids which are the pleomorphic rhizobia. This is the mechanism of symbiotic nitrogen fixation in rhizobium. Now coming to the biochemistry of nitrogen fixation in the nodules. So I have already told you in the previous classes that there is an enzyme called nitrogenase which is important, or which is responsible for conversion or fixation of nitrogen into ammonia. Now nitrogenase is a very sensitive enzyme. It is sensitive to oxygen, and even a small amount of oxygen can disable the enzyme or make the enzyme inactive. So nitrogenase is present in case of rhizobium. Nitrogenase enzyme is present within the bacteroids and it is not released into the plant cytoplasm. So whatever oxygen is present in the plant cytoplasm. It is not present inside the bacteroid, or it is not present inside the symbiosome. So you can see over here, this is the symbiosome membrane. You can see this blue color, that is the symbiosome membrane. Inside the symbiosome, we have the bacteroid. So this outer layer is the bacteroid membrane. Inside one symbiosome, there can be multiple bacteroids. Here I have just shown you the example of one bacteroid inside one symbiosome membrane. So the bacteroids, now because oxygen is not being allowed inside that easily, so the organic compounds are transported across the symbiosome membrane, especially succinate, fumarate and malate. So whatever the bacteroids need or they require for energy, that is being provided by the plant. So the plant will do photosynthesis as a part of its regular cycle. After photosynthesis, it will form sugars. These sugars will be converted to organic acids with the help of Krebs cycle and other cycles. And these organic acids, mainly succinate, malate and fumarate, these organic acids are then transported through the symbiosome membrane for the usage by the bacteroid or usage by rhizobium. So the bacteroid is depending on plant for the energy source that is going to help in nitrogen fixation. All of these organic acids are essentially energy source so that nitrogen fixation can happen. Now let us see how that works. So these organic acids, now once they enter inside, either they will enter into citric acid cycle of the bacteroid or Krebs cycle of the bacteroid, or these organic acids will form pyruvate. Either one of these can happen. At times the organism wants electrons, or other times it is wanting energy. I'll tell you how it works. So these organic acids provided by the plant, once they enter the symbiosome membrane or once they are transported across the symbiosome membrane, these organic acids, if they enter the Krebs cycle, from the Krebs cycle we have release of NADH2 and FADH2, basically reducing equivalents. These reducing equivalents are now going to enter the electron transport chain. So the NADH2 molecules and FADH2 molecules will provide electrons to the electron transport chain which is present on the bacteroid membrane. These electrons which are provided by NADH2 and FADH2 travel through the different electron carriers and are finally taken up by oxygen. Oxygen accepts these electrons and gets converted into water. In this process, we have formation of ATP from ADP. This ATP is the energy source that is required for the actual process of nitrogen fixation. So, that is one route in which these organic acids can be used. Otherwise, these organic acids can form pyruvate, and pyruvate can directly give the electrons to nitrogen. The biochemistry or the biochemical reaction of this I have already shown you in my previous video lectures. Please do refer that. So, pyruvate provides these electrons to the nitrogen, and in the presence of nitrogenase enzyme and ATP, nitrogen is being fixed into ammonia, or nitrogen is being converted into ammonia. This ammonia, so assimilation of ammonia, now once ammonia is being formed, this ammonia has to be assimilated, or it has to be fixed, or it has to be accumulated in the plant. So this ammonia which is formed can be used to form amino acids for the bacteroid itself, or most of the ammonia is transported back into the plant cell so that the plant can assimilate it. So though the bacteroids are able to assimilate some of the ammonia into organic form, the levels of ammonia assimilatory enzyme in the bacteroid is less, or they are low. So most of the ammonia is now transported back into the plant cytoplasm, where through different pathways they are being converted into either amides or urides. Now we have glutamine synthetase pathway or glutamine synthetase enzyme which will assimilate the ammonia that is coming from the bacteroid into glutamine and asparagine, which are known as amides, and urides, which are basically allantoin and allantoic acid. These are then used by the plant to convert into various amino acids by the respective pathways. So this is how the nitrogen fixation occurs inside the nodules. When we talk about biochemistry of nitrogen fixation, you need to show about the way nitrogenase is converting the energy or the electrons that are released by pyruvate, how the two subunits of nitrogenase enzyme, basically ferredoxin is involved as an electron carrier, and the two subunits, that is Fe protein and MoFe protein, are taking the electrons and giving it finally to nitrogen and converting it into ammonia. This is the general reaction of biochemistry. But in case of root nodules, you need to know about how the symbiosome membrane is helping in making sure that oxygen is present only in the plant cell and is not interfering with the nitrogenase enzyme present inside the bacteroid. Now when we talk about this root nodule, so you can see over here we have presence of a molecule called as leghemoglobin. Leghemoglobin stands for leguminous hemoglobin or legume hemoglobin. The legume hemoglobin works like a regular hemoglobin, so it is an oxygen carrier and it has a heme protein, so it is red in color. That is why if you see young nodules or active nodules, they will be pink in color because of the presence of leghemoglobin. So this leghemoglobin will take the or sequester or scavenge the oxygen which is present and form a complex, that is oxygen leghemoglobin complex. This complex will release the oxygen in a slow or in a stable manner such that too much of oxygen is not present in the vicinity of nitrogenase. So it is only providing oxygen when required. That oxygen will immediately take up the electrons and convert into water, so that there is no presence of oxygen in the vicinity of nitrogenase. This is being done with the help of leghemoglobin. Let us study more in detail about that. So when we talk about a root nodule, generally the type of root nodule that is present in legumes is of the indeterminate type, or when we say what type of root nodule it is indeterminate type, because it maintains an active apical meristem or an active apical zone which is continuously dividing, forming new cells when throughout the life of the nodule. So that type of a nodule is called as an indeterminate type of nodule, where there is an apical meristem which is continuously dividing to form new cells. In the nodule, the central zone is where what I just showed you, in the central zone, that is where we have the actual nitrogen fixation occurring, or that is the actual nitrogen fixation zone where leghemoglobin is present, and that is why it is a pink color like I told. The area of nodule which is present towards the root is called as the senescent zone. Senescent zone stands for dying zone. Senescence is basically death or dying. So senescent zone is where the plant cells and their bacteroid contents are being degraded. So due to breakdown of the heme component, usually this area is seen as a visible green color. So the pink part is only over here in the central zone, because that is where we have active nitrogen fixation and we have presence of leghemoglobin which is imparting the pink color. Now this leghemoglobin, so why does the organism need this leghemoglobin? Like I told, nitrogenase is extremely sensitive and it needs to, or the plant and bacteroid need to make sure that there is no oxygen at all in the vicinity of the nitrogenase enzyme. So leguminous plants have two mechanisms. The major mechanism is leghemoglobin, or it is also called as legoglobin. Now this is an oxygen carrier with heme protein in it. That is why the red color is being given, due to the heme protein. Initially it was thought that the heme moiety or the heme part is from the bacteroid and the globin part, globin is actually a protein, so the globin peptide was from the plant. This is how it has been believed to be for many years, that the heme part comes from the bacteroid and the globin part comes from the plant. But actually speaking, new research shows that it is the plant that produces both the components. So roots which don't have, if they are not colonized by rhizobium, they don't synthesize leghemoglobin. The plant needs to be colonized with rhizobium, but both the parts, heme and globin part, are produced by the plant itself. This is what new research shows. So this is a protein which is having a molecular weight of 16 kilodaltons, and what is its purpose or function? It is buffering the concentration of oxygen, which means it is providing oxygen in a very sequestered manner, or it is providing oxygen in a controlled manner for ATP production. It is not providing all the oxygen together. It makes sure that it holds on to the oxygen and releases it slowly. So it buffers the concentration of oxygen which is present in the cytoplasm of the plant cell, and oxygen required for the aerobic respiration to produce the ATP that is required for nitrogen fixation. Because if you remember, nitrogen fixation is extremely costly in energetic basis, extremely energetically costly, because it requires a very large number of ATPs. 16 to 18 ATPs are required for one molecule of nitrogen. So oxygen is required because more energy is required, but that oxygen should be released slowly, or we call it as it buffers the concentration of oxygen that is present in the cytoplasm of the plant cell. And nitrogenase is oxygen sensitive, so leghemoglobin maintains that the concentration will be low, concentration of oxygen will be low, just to allow nitrogenase to function, but not too high that it can stop. So nitrogenase is functioning, aerobic respiration is happening, and ATP production is happening. That is what is the purpose of leghemoglobin. This is an extremely important compound, and without the presence of leghemoglobin, the organism will not be able to, the plant will not be able to have any fixed nitrogen. Even if rhizobium is able to go inside, if there is no leghemoglobin, the plant will not be, the organism will not be able to fix nitrogen. So if you see a nodule, this is what I was telling you about. In the nodule, this is the bacteroid infected tissue or the symbiosome. If you can see here, in one symbiosome, there are several bacteroids, or there can be just one bacteroid, that depends. This is called as a symbiosome. So apart from leghemoglobin, so root nodules have another mechanism to make sure that they protect their nitrogenase, that is by presence of hydrogenase enzyme. Now hydrogenase is the enzyme which is removing oxygen from the vicinity of nitrogenase, because hydrogenase converts oxygen, it combines it with hydrogen to form water. So in both the ways, the nitrogenase enzyme is being protected. One is by the presence of leghemoglobin. This is the major reason why nitrogenase is not affected, and the other one is the hydrogenase enzyme. Both of these make sure that the oxygen that is present only in the plant cell. Whatever is the concentration of oxygen, it does not enter the symbiosome, or it does not enter the bacteroid in a rush. it is given slowly or it is buffered and given such that the aerobic respiration keeps occurring but nitrogenase is not being affected. So this was about the mechanism of nitrogen fixation in leguminous plants or the symbiotic nitrogen fixation. Please remember, you need to know about the mechanism or the steps in detail. Then you need to talk about the biochemistry. Biochemistry is the reactions are same as I have taught you before, but in this case, in the case of symbiotic nitrogen fixation, additionally what you need to write is the slide or is the previous slide where I had shown you the symbiosome membrane and how the organic acids are entering from the plant and either being used for electrons or being used for ATP. So that also has to be mentioned. And you need to write about the mechanisms by which the root nodule protects the nitrogenase enzyme from oxygen. There are two mechanisms there. One is presence of the protein, that is leghemoglobin, the oxygen carrier, that is leghemoglobin, and the presence of hydrogenase enzyme. I hope you all have understood today's class. Thank you.